Good evening, everybody. Welcome to Brain School Evening 2. I see a lot of the same faces. I see a couple faces I didn't see last night, but who's counting? I'm looking very carefully. We're missing all the kids we had in the back row. They must have been here for the snakes last night. Okay, well, welcome back. It's great to see everybody again. You're in for a very exciting evening. We're going to follow the same format we did last night, but let me just remind you, we had the virtual reality Explore the Brain exhibit set up in the lobby for those of you who had a chance to see it. One of our graduate students, Ubada Sabag, was down there demonstrating it. Who got to see that? Raise your hand. Okay, good. Fair number of people. And tomorrow night we will have Brains in a Bucket Part 2. Dr. Fox will be out there once again to let you handle human brains and find your way through the brain and so forth and experience the somatosensory and visual cues from that. And then on Thursday evening after the last class, we'll be having a group from the Alzheimer's Foundation who will be putting on an interactive display out in the lobby as well. So there's still a lot of good things to come. Hopefully the rattlesnakes last night didn't scare too many people away. It doesn't look like we've lost a whole lot of people. So it's my pleasure tonight to introduce the program. We will have two speakers again, as we did last night, presuming that they both get here within the amount of time. And we're going to start off with Dr. Mike Fox. Dr. Fox directs the Center for Translational and Developmental Neurobiology next door in the Fralin Biomedical Research Institute. And he's an expert on understanding how visual systems in the brain get put together and how they function, how the various molecules there play a major role. And then he will be followed by Dr. Edmundo Rubio, who's the Chief of Pulmonary and Critical Care Medicine at Carilion Clinic. And Dr. Fox will start out telling us about what frogs have taught us about rhythms and blues, teaching us about circadian rhythms and how those operate in the brain. And then that will be followed by Dr. Rubio telling us about sleep problems in modern times. So without further ado, it's my pleasure to introduce Dr. Mike Fox. Mike, it's all yours.
Thank you, Mike. Can everybody hear me in the back? All right, thank you all for coming tonight. I am going to start off by doing kind of like the cardinal sin in giving a talk. And that's, I'm going to change my talk title, I'm sorry. But just talking about frogs wasn't enough. So I'm going to talk about what frogs, flies, and furry things taught us about our rhythm and blues. So last night we had some great examples of how nature and animals have evolved to adapt to their environments, adapt to survive, and even in some cases thrive. Today we're going to kind of shift from that or build on that and talk about how organisms, many organisms on planet Earth, have evolved not only to adapt to their environment, but to predict changes in it. And this is a really important function of them, to predict changes in seasonal changes, to predict lunar changes, to predict tidal changes, and what I'm showing you here, to predict changes in the day-night cycle. And being able to predict and adapt to these changes is an important biological function of these organisms. And today we're going to get into the mechanisms, how organisms can do that last one, which is adapt to changes in the day-night cycle. And we call this the circadian rhythm, which is really relating to any biological processes that recur naturally on roughly about a 24-hour period. And what's really important is that this is an intrinsic ability of organisms. It doesn't take environmental stimuli to be able to have this circadian rhythm. And of course here I say it happens even in the absence of light fluctuations, which is the day-night cycle, but it happens in the absence of temperature changes. It happens in the absence of lunar changes or tidal changes or lots of other changes that occur. And so this is an intrinsic ability of organisms. And throughout this talk, I'm going to try and highlight some animal models that we use to study it, both in the lab and in our environments. And so I'm going to start with some of the furry creatures. The furry creatures have taught us a lot about circadian rhythm because they have this very innate ability to entrain their activity, and we can see that very easily. And so the furry creatures I'm going to talk to you about are shown here. This is a golden hamster or a Syrian hamster, and this is, of course, a mouse. And anyone have any of these? Yeah, a few? Okay. So you probably, those that raised your hand probably know this innate activity is if you put one of these critters into an environment that has a treadmill that looks like this, they go forever during their active period. For those that didn't raise your hand, I'll give you an example. You've probably all been to the YMCA or the RAC or Gold's Gym, and when you walk in sometimes you'll see someone on a treadmill going, and you'll do your exercise and you'll be exhausted and you'll leave, and that darn person is still on the treadmill. And that's essentially these little critters. When they get on a wheel during their active period, they can run 2 to 5 kilometers a day. And as a scientist, we can easily track that by counting the number of times this wheel rotates, and we can chart that into what we call an actogram. And I'm going to show you lots of actograms today, so let me walk you through this one. So here on the top, we plot time, and here you can see time 0, which is really the time when lights come on. And so you can see there's kind of gray bars here and under here, and then white here. And so this designates dark periods and light periods. And then you can see there's all these rows, and each row is a day. And within a row, you'll see these little black blips, and those black blips are wheel rotations. And the larger the black blip, the more wheel rotations. So I think what stands out is that mice, rodents, when they're in captivity, entrain their activity to just 1/2 of the day. And it turns out they do it when the lights are off, so we call them nocturnal. And they have very little activity when the lights are on. And so this has been really important for us to be able to manipulate and study what can modulate or change behavior like circadian behavior. Now, this isn't something that's only in rodents. I'm pretty sure that many of you might be wearing one of these. And it does more than just buzz when you haven't gotten up from your desk in 40 minutes or an hour. It actually tracks your steps over days, weeks, months. And so you can download that data tonight, and you can make your own actogram, and it would look something like this. And you'll notice now it looks very similar to the actogram I just showed you for the mouse. So here you have a month of activity. Here you have each day of activity. And if you follow this all the way down, you'll see the times. And this individual isn't very active at night or in the mornings. Gets up about 8 a.m. and is quite active all day, sometimes until about 1 or 2 in the morning, and then goes through another period of inactivity. And so this is their circadian rhythm. So it's not just our activity that can be entrained to this biological clock, but other processes as well. And so here I'm showing you just a few. The top one that you see there is temperature. And what you'll see is on the day-night cycle of a human, during the day periods, our temperature is about 1 degree Celsius or 2 degrees Fahrenheit higher than during the night periods. Now this, like activity, is kind of a whole body readout of our circadian rhythm, but we can see circadian rhythms even in individual cells that, say, produce hormones like growth hormone or cortisol, which is a steroid. You can see that they also have rhythmic activity, where they're produced only during certain periods of the day-night cycle. So if we start here with cortisol, you'll see it's highest when you wake up, and it decreases during the day to its lowest point when the lights go off, and it rises quickly overnight and then does that again. To me, what's most interesting is the growth hormone here, which is very flat during the day and peaks kind of early in the night, but it starts to peak, or it starts to rise before the offset of light. And so that kind of reiterates what I said earlier, that organisms have evolved a mechanism to predict those changes in the environment, like lights going off, so that they're better prepared for whatever function they need to do. So I think this leads us to, how does this happen? How do our bodies have these rhythms? And at an individual cellular level, how does each of our cells have this intrinsic rhythmicity? And people have studied this from a cellular and molecular mechanism for about 50 years. And about a year and a half ago, the Nobel Prize was awarded to these 3 scientists for their discoveries or their contributions to the molecular mechanisms that control circadian rhythms. And really the star of the show for these 3 gentlemen was not a furry critter, but this critter. And this is a fruit fly. Fortunately, it's not to scale with these guys right now. It's much smaller. And just like the furry critters, and just like you, it has circadian rhythms. And we can see those circadian rhythms in an actogram that's of a fly. And this is an actogram from a fly. It shows the same information that I've shown you for the mouse and the hamster and for the Fitbit. It's plotted a little differently. And so here we're plotting just activity based on hour of a day. It's one day. And every day gets averaged into this, so you can see there's standard deviation on all these bars from the averaging of day 1, day 2, day 3, day 4. And then here on this axis, we have the amount of activity. So what should stand out is rather than having all of its activity in the night or all of activity during the day, it's bimodal in its activity, and it's very active when the lights come on in the morning, shown here. And it's very active when the lights go off in the evening, shown here. And that's very typical of insects, mosquitoes, things like that. So you should concentrate your outdoor activity right here. So this organism, just like the furry critters and just like us, has circadian rhythmicity. So that really wasn't what made it such a good tool to study, to identify genes involved in circadian function. What made this really amenable to identify these genes was it's the genetics involved in this organism. And so I'm going to walk you through like 101 for fly genetics. What makes this such a good model to identify genes? It's a technique called forward genetic screens. You apply a forward genetic screen if you have no idea of any genes involved in a biological function you're interested in, say circadian rhythm. So you have no leads or anything. You have to identify genes that might be important. So what you can do is induce spontaneous mutations in a fly by giving it some sort of drug. This is actually an alkylating agent, but it induces changes in the DNA that we call mutations, which are designated here. And then you can cross or mate that fly, which is a male, to a female fly, and you get offspring that carry that mutation. Now, just like us, these flies have 2 copies of every gene, and so they have 1 copy of that mutation after that first cross. You then separate literally thousands of flies, and you do it again. And you keep crossing them until eventually you're crossing one of these light pink flies to another light pink fly, and you end up with some light pink flies which have 1 mutation, kind of boring, and some dark pink flies which have 2 mutations, which is now going to be penetrant, and you're going to see a phenotype. But you have thousands of these, and you now start looking at their behavior. And you only care about one behavior, circadian behavior. So you're measuring their activity over a 24-hour period. And lo and behold, you identify some mutant flies that don't have this circadian behavior, and bam, those are the ones that you're going to study. And they don't have gene names yet. So you get kind of creative, which is what people who study flies do, and they come up with crazy names. Some of you may have played a video game called Sonic Hedgehog. That was from one of these screens where the hedgehog was where the fly was extremely fast, and so they called it Sonic Hedgehog. In the case of circadian biology, they came up with some pretty cool names, which are shown here. So these are the names of the fly mutants: period, clock, timeless. Are you getting the gist of what they were interested in here? Double time, and there were others. And so then they had to identify what these genes did. And this is really a tough question. You have genes, you don't know what they do, and you think they play a role in giving a cell an intrinsic ability to count time. And it turns out all of these genes play a role in transcription and translation. And these are fundamental processes of a cell where you go from DNA to RNA. That's called transcription. And then when you take that RNA, you move it outside of the cell nucleus and to a ribosome where it can be translated into an amino acid chain and then folded into a protein. And that process is called translation. Now it turns out this fundamental biological property takes 6 to 12 hours. Well, that's a pretty useful time period if you're trying to generate a clock inside a cell that's going to be about 24 hours long. And so after about 20 years, those 3 gentlemen that I showed you pictures of before came up with a hypothesis and tested it, that there was a loop of this transcription translation event, that each side would take 6 to 12 hours, One side would generate one set of these factors. We call these transcription factors because they're important for taking DNA and turning it into RNA. And the other side of the loop would do the same thing but block the first ones. Each side taking about 12 hours. Now we have an intrinsic clock for a cell that's about 24 hours long. Now it's kind of inaccurate because in the fly, it's only 20 hours long. And in the mouse I told you about and the hamster, it's 23 and a half hours long. And in you, it's 25 hours long. So all of our cells have this intrinsic clock, but they're all a little different. And that's actually really important because we want to be able to adapt to our environment. And we want to be able to take in some environmental cues and shift this clock forwards or backwards a little bit. And we call this entrainment. And I'll show you an example of that with our furry critter again. So here's our mouse. And here is an actogram from a mouse that went on for about 45 days. And at the very beginning, you can see it has normal night-dark cycle. So here's night, here's light, here's night, here's light. And then after 15 days, we decided to put this mouse in total darkness. I could do this to you if you'd let me. You'd have to give me your devices, your watch, anything, any association with the outside world, and you just let me put you in a dark room for 45 days, and I could make an actogram that looks just like this of you all. So when this mouse went into total darkness, you'll see every day, without an external cue, it thought it should start exercising half an hour earlier, half an hour earlier, half an hour earlier, half an hour earlier. We call this free running. And it exposes an animal's intrinsic circadian clock. If you actually did let me do this to you, you would free run the other direction because every day you would run 1 hour later, if you would actually get on a wheel, which I don't know whether that would happen. Now, that shows you the intrinsic clock. So now I want to show you that you can re-entrain to light and dark cycles again. And that's shown at about day 30 when we decide to put the lights back on again. And you can see there's some kind of errors in their running the first few days, but eventually they re-entrain to a 24-hour cycle in the presence of those light cues. And I want to point out, a lot of times we give these talks and we basically steal pictures from textbooks, but a lot of these were taken actually here. A lot of these graphs were done here at the Fralin Biomedical Research Institute. And so I want to give credit to the student who did this. Gabi, who's probably in the back in shock that I'm showing her picture now. So sorry, Gabi. But I wanted to actually show some data that was collected here at the Institute this year. So one of the things Gabi did after she did this experiment with some mice was she wanted to show another way that mice entrain. And we call this a jet lag experiment. And so she took these mice, which are photo entrained here. You can see all their activity starts as soon as the light goes out. And then she shifts the light on, light off period by 6 hours. And you can see it takes about 3 days for the mice to re-entrain. And as soon as they do, she's kind of cruel, so she changes it back, and then they have to shift their clock back. This is essentially like putting the mouse in a box and sending it to Paris without all of the benefits of being in Paris. And of course, all of us are kind of experiencing this right now. 2 days ago, we shifted our clock by a meager 1 hour, and for days we've been complaining about it because of how much it messes up our system. So fear not, just another day or two, and it'll be all normal again. All right, so how does this happen? How do we photo entrain? How do we take light-derived cues and readjust our circadian clock? And we do it with a specialized tissue in the back of your eye called the retina. It's actually part of your brain that ends up in the back of your eye. And light comes through this retinal tissue, and it activates a bunch of cells which we call photoreceptors. You've probably heard of these. These are called rods and cones. And those rods and cones detect that light and turn it into an electrical signal that they're going to send through a bunch of interneurons here in the middle of the retina. And then eventually those interneurons are going to activate a type of projection neuron, which sends a long axon into the brain, probably my favorite cell in the whole brain, the retinal ganglion cell. And this is how light goes from how we detect light and how we send that information into the brain. So where does it go? Now, you've heard yesterday about few of the projections from the retina to different brain parts. We're going to ignore all of that, and we're going to focus on one itsy-bitsy, teeny-weeny little part of the brain right here in the hypothalamus it's a terrible name, I'm sorry, Suprachiasmatic Nucleus, or SCN. So this SCN in you all is about 50,000 neurons, which sounds like a lot when I say it right now. But when you think that you have 86 billion neurons in your brain, it accounts for 0.0000006% of the neurons in your brain. And yet, that tiny little fraction of neurons controls all of your circadian activity in all cells of your body. It is the master regulator of circadian functions. And it's that tiny little place there. So how does it do it? It has a few mechanisms where it can kind of talk to all the cells in your brain and all the cells in your body. I'm just going to mention one, which is it sends connections. So here's the SCN. It sends connections or axons into the brainstem, which then are relayed back up into this little gland here called the pineal gland. And the pineal gland produces melatonin. When it's activated, melatonin goes down, and when it's turned off, melatonin goes up. And melatonin, the balance of or amount of melatonin is important for your sleep-wake cycle. So if you disrupt that, you'll disrupt your sleep-wake cycle. You'll have mood problems. You might get seasonal affective disorder. And so this is how you go from light to the SCN, suprachiasmatic nucleus, to the pineal gland, to melatonin production. Now, it's not quite that simple. So if you disconnect the retina from the brain, everything I just told you doesn't work. And here's an example of that with a mouse that we have here that has no connection between the retina and the brain. And you can see it's free running. Doesn't matter if you're in light or dark. This mouse is just showing its intrinsic rhythmicity, and it cannot photoentrain because no signals go from the retina to the brain. The same is true in humans with diseases that affect this cell, like glaucoma. And in humans, we would call free running something like non-24 syndrome, where you can't photoentrain. But it turns out there's other retinal diseases which make you blind, which don't disrupt your circadian clock. And so one example is shown here. This is a 23-year-old patient that has defective rods and cones, but his ganglion cells were fine. And you can see when he self-reports his sleep patterns that he still is pretty much photoentrained, even though he can't perceive light and he's legally blind. And if you give him a very bright flash of light, which he can't detect, it changes his melatonin production in his pineal gland. So this was really puzzling to us for a long time. And who came to solve this question? I know you're all going to get it right. It's the frog I started talking about at the very beginning. It's not a regular frog. It's not like your cute find in your backyard frog. It's kind of this weird live underwater in Africa frog called the Xenopus laevis. And Xenopus stands for strange foot, which you can see kind of has some strange feet. It's called the African clawed frog. And it uses these to dissociate food and put it in its mouth because it doesn't use its tongue like most frogs. It's actually illegal to own these here because they're such voracious hunters that if they got loose, they would just eat all the other organisms in any lake that they got into. And so you can use them for research, but you can't have them at home. And of course, the claws aren't what I want to talk about. What I want to talk about is you can take these eyes off of this frog, and it will still be able to detect light. And so a researcher who's now at UVA, Iggy Provencio, found that it has certain cells on its skin that have an opsin, which is a type of protein that detects light, just like the opsins in your rods and cones. And he called this melanopsin, and it has a very similar structure to the opsins in rods and cones, which I didn't say it before, but which we call rhodopsin, and then these three different conopsins, which are also called photopsins. Now, in the retina, all three are present. All three are present in the frog retina. All three are present in your retina. But it turns out they're made by different cells. So, of course, rhodopsin and conopsins are made by our rods and cones. What was really surprising to us 18 years ago was that melanopsin is not made by rods and cones, but it's made by a very small set of ganglion cells, which are those projection neurons which go to the brain. And by expressing this opsin, now these ganglion cells are actually photoreceptors. And so we call them intrinsically photosensitive retinal ganglion cells because as scientists we like really big confusing words. And so that's shown up here. And here's a picture of them from mouse retina. It's viewed as if you're down here looking up at the retina, so the retina is folded out. And in this image, which is about 1 millimeter by 1 millimeter, you should see thousands of ganglion cells. But when we use antibodies against melanopsin, you only see about 15. And so we know that only about 2% of your retinal ganglion cells make melanopsin and are photosensitive. And in this image, I'll just highlight one. Here's the cell body of one of those intrinsically photosensitive retinal ganglion cells. And these are the dendrites which stick out. That's going to be what receives information. And then this is its axon. And you can see its axon is going to go to this hole, which is where it leaves the retina and goes to the brain. And it turns out, you can probably guess at this point where this axon is going to go. It goes to the suprachiasmatic nucleus. And I know this is a mouse version of a brain, and I haven't shown you that, but just trust me, this tiny little thing is the suprachiasmatic nucleus, and here it is where we've labeled those intrinsically photosensitive retinal ganglion cell axons. And sure enough, if you take away melanopsin from a mouse, or if you kill these cells specifically, the mouse loses its ability to photoentrain. You can see that this same cell projects to a few other places in the brain, and I'll be honest, we don't know what a lot of those connections do yet. But about 3 weeks ago, a paper came out about a projection of one of these intrinsically photosensitive retinal ganglion cells to the thalamus, and they kind of traced the circuit of this, and they found that this same cell, a cell that makes a protein that we identified in a frog, was important for ameliorating depressive symptoms. And they think that this might be a mechanism how light therapy might be useful for treating things like depression. And so, again, I think this is a perfect example of how what we learned in a frog told us some fundamental biology about the retina, and then could be applied to how we understand our rhythms and our blues. And so I know I've talked about a lot today, but here's the take-home message. If you forget everything else, that's fine. Just remember what I have on this slide. What have frogs taught us about rhythms and blues? Well, frogs, as I said, gave us melanopsin to study. And melanopsin, what I haven't told you yet, is extremely sensitive to a certain type of light. It turns out it's very sensitive to blue light, and blue light has therefore a strong effect on our circadian rhythms and our mood. So why is this important? Well, it turns out before we identified melanopsin, a lot of times we used blue light at night. We thought it had a calming effect. Hospitals used it in their hallways. Nursing homes used it in their hallways. We used it as our night lights. A lot of us probably have clocks which have blue numbers on it at night. Well, that is a very strong inducer of our circadian clock. It depresses our melatonin secretion, and we want to be awake when we see blue light. And of course, these are just some examples, and you're probably all thinking like, I don't have a Batman light anymore, I'm good. I'm done with Pac-Man or unicorns. Well, it turns out blue light comes from many sources, and I'm guaranteeing that all of you look at this thing right before you go to bed, and there's a lot of blue light that comes out of our devices. So if you take anything from this talk, what frogs have taught us about our rhythms and blues, it's don't get too much blue light at night. And with that, I'm going to end my talk, and after our next talk, I'm happy to answer any questions.